The broadcast is now starting. All attendees are in listen-only mode.
Good morning, everyone, and welcome to the Internal Phosphorus Loading webinar offered by the New England chapter of the North American Lake Management Society. My name is Amy Smagula, and I will be moderating this session today. Just a bit about who we are. NALMS is an affiliate of the North American Lake Management Society. As our name implies, we're comprised of members from the 6 different New England states, and we have a diverse membership of citizen scientists, professionals in various fields of lake management, members from academia, students, agency personnel, and more. Due to the pandemic, we decided not to host an annual meeting this year. Instead, we are offering webinars to bring you guys in and share information. This is our second webinar. We had one in April on shoreline plantings, which is available online. A little bit about our parent organization. The mission of NALMS is to forge partnerships among citizens, scientists, and professionals to foster management and protection of lakes and reservoirs for today and tomorrow. We have about 1,000 members in NALMS, mostly in North America, but some international. NALMS is celebrating its 40th anniversary this year, and we are also hosting an annual meeting from November 16th through the 20th. We have almost 200 presentations and more than half a dozen workshops that we'll be offering. Registration is open, and you can access registration on the NALMS website. We really do hope that you'll join us for some or all of the NALMS symposium. About today's webinar, we are talking about internal phosphorus loading. We will be on for an hour and a half. We have 3 20-minute presentations with a block of about 15 or 20 minutes at the end for questions. I will be moderating the question and answer session. All of the participants are on mute. However, you have the option to use the chat function or the question function to pose your questions to the presenters, and I will read those out and the presenters will answer those questions later on in the webinar. We are recording this webinar, and it will be shared via a YouTube link off the NALMS and the NALMS websites. And I am also going to email a link to the video of the webinar to each of the people that are registered for the webinar, probably in a day or two. Our presenters for today's webinar include Dr. Alan Steinman from the Annis Water Resources Institute at Grand Valley State University. He is going to open us up and talk about understanding internal loading. Next, we'll hear from Dr. Ken Wagner with Water Resource Services. He is going to talk about the link between internal phosphorus loading and cyanobacteria blooms. And then we'll hear from Dr. Mike Lurling from Muskegon University, and he is going to talk about controlling internal phosphorus load in lakes. So that we can let our presenters roll from one to the next, I'm going to share their biographies in advance. So first, starting with Dr. Alan Steinman. He holds a doctorate in botany and aquatic ecology from Oregon State University and a master's and a bachelor's degree in botany from the University of Rhode Island and the University of Vermont, respectively. He works in the Alan and Helen Hunting as a director of Grand Valley State University's Annis Water Resources Institute, and he's held that position since 2001. Before that, he was in Florida, and he worked as the director of the Lake Okeechobee Restoration Program. Dr. Steinman has a number of scientific publications to his name, as well as a lot of awards and recognitions. He also currently serves as the associate editor for the journal Freshwater Biology. Our presenter, Dr. Ken Wagner, holds degrees from Dartmouth College and Cornell University. His PhD is in natural resources management, and he has over 40 years of experience working on lake assessment and rehabilitation. In 2010, Dr. Wagner started a consulting firm, Water Resource Services, where he focuses on water supply protection and lake management consulting. Dr. Wagner is the former president of the North American Lake Management Society and also the former editor-in-chief of Lake and Reservoir Management. And then Dr. Mike Lurling holds PhDs from a PhD and a Master of Science from Wageningen University in the Netherlands. He has over 25 years of experience in aquatic ecology, water quality research, and university teaching. He works as an associate professor in aquatic ecology and water quality management at Muskegon University, and is a senior researcher at the Aquatic Knowledge Center at Muskegon. He is also chair and on the board of several international working groups related to different topics in limnology, including plankton ecology and cyanobacteria. And he too also has a number of publications in peer-reviewed journals and in scientific reports. So now we're going to transition over to our first presentation with Dr. Alan Steinman, and he is going to talk about understanding internal loading. And I am just going to switch over to his screen. So just give us a moment.
Thank you, Amy. Appreciate the opportunity and the invitation from you and Ken to present on understanding internal loading today. A lot of the information that I'm going to present comes from this book that Mike, who's going to present third, is holding with Gertrude Nurnberg, and then Emily Kindervater, and that's Brian Spears, who co-edited the book with me, and that's me in the middle there. The presentation will consist of 5 main areas. First, talk a little about definitions and what internal loading actually is. Then sources and mechanisms of internal loading, how to measure it, and its relative importance compared to external loading coming from the watershed and outside the lake. And then finally, finish up with knowledge gaps and recommendations. This is the definition that we developed, that Brian and I developed from the book, where it can be generally considered as all physical, chemical, and biological processes by which the P is mobilized and translocated from the benthic environment. But there have been other definitions and other caveats associated with internal phosphorus loading. For example, Hupfer and his colleagues suggested that it only should refer to net sources of P that last for more than 1 year. And then Orahel and her colleagues said that it should be restricted to the phosphorus leaving the sediment that reaches the overlying water column and results in cyanobacterial blooms. And to a large degree, these are academic discussions. If you're having internal loading in your lake, you don't really care about the definition. You want to know why it's occurring, how it happens, and how you can control it. And hopefully over the next hour, you will get that kind of information. What we did was identify 4 major classes of sources for internal loading. One is biological, chemical, physical, and then this combination of bio and physical. And I'll go into this in a little more detail in subsequent slides. But very briefly, for the biological, we use an example here of burrowing invertebrates. The bioturbation basically disturbs the sediment, they rework the sediment, and as a consequence, as the sediment and organic matter gets mineralized, phosphorus ends up being released from the sediment into the water column. The chemical is probably the best known process that goes on, associated with iron reduction, which releases the phosphate ion, which then can diffuse out of the sediment into the water column. Physical is where there might be wind wave action, or if you're in a shallow lake, it could actually be prop action from boats that disturbs the sediment, resuspends it up into the water column, and then you either have increased amounts of total phosphorus, or the phosphate ion may desorb from the particles and end up becoming bioavailable in the water column. And then finally, you have this biological physical interaction, where in this case, benthic feeders like gizzard shad, common carp. If you're over in Europe with Mike, you might have bream that's responsible for disturbing the sediment and having the same reaction as we saw over here to the left. Some other examples of these biological factors that can influence internal loading. Well, these chironomids or oligochaetes in lakes that perhaps have eutrophied systems are building the tubes and burrows. They rework and ventilate the sediment. As that oxygen gets into these burrows, they basically aerate these tubes and allow the aerobic respiration to go on by microbes, and that will break down the organic matter and release the bioavailable phosphate ion. Cyanobacteria, as we all know, also vertically migrate thanks to their gas vacuoles, and then so they can take phosphorus from the Hypolimnion up into the Epilimnion and vice versa. And then as organic matter decomposes like bacteria in lakes as a natural process, the phosphorus inside the bacteria are released, and that can then get into the water column as well. So let's take a look at a flow chart associating with the impact of bioturbation, where we have these chironomid larvae in the sediment. And here to the left, they're basically reworking, they're building tubes and they're disturbing the sediment, they're increasing the particle mixing, and as a consequence, you're going to get more mineralization of this organic matter. To the right, in the burrows, they're bioirrigating, they're basically sending oxygen through these systems. As a consequence, that oxygen increases the aerobic respiration from the bacteria and microbes. In both cases, you have solutes that are being released and eventually diffuse out of the sediment and back up into the water column, where it can then stimulate algal blooms. Chemically, again, that most common process, which I'm actually going to take you through in a cartoon in the next slide, so I'm not going to spend much time here on the redox-driven release of phosphorus from iron. Also, there can be desorption of phosphorus bound to mineral surfaces. This is usually pH-driven. High pH, you can release the phosphorus associated with calcium, and therefore make that bioavailable P. There's also, as some of these minerals crystallize, they age over time, and as they age, they end up releasing and dissolving the phosphorus that might be attached to elements like calcium, iron, manganese, or aluminum. And then finally, probably the least common process that we see is the phosphorus complexes associated with humic substances. As they dissociate through this indirect binding, that phosphorus can be released as well. So in this cartoon, this very simple cartoon, we have our lake with the water on the surface, the sediment on the bottom. And in the water column, we have the bioavailable or soluble reactive phosphorus that's available, which of course is taken up by the algae and forms particulate phosphorus. Now, some of it can be re-released back to the water column, but by and large, this reaction moves from left to right. The algae ultimately die in the fall. They sediment out onto the sediment, where they can either be resuspended or they can decompose. And as they mineralize, that bioavailable phosphorus now enters into the pore water of the sediment. Now, if your lake is oxygenated at the bottom, it might be polymictic or it may be turning over, the iron stays in the oxic form, in the Fe3+, or the ferric form, and in that oxygenated form, it binds to phosphorus and it precipitates out and it stays there. It's no longer bioavailable. But, for example, in lakes that stratify, and in the summer months, we know the bottom waters go anoxic. Now that phosphorus, the iron, is chemically reduced to the ferrous form, and it releases the phosphorus, which then can diffuse out into the water column and start this process all over again. Of course, it's possible to stop this diffusion by adding chemical inactivants like alum or phoslock that are not redox sensitive, and I'm sure Mike will talk a little more about that in a few minutes. Now, this process doesn't account for sulfate, and I've added sulfate into this, particularly since this is representing the New England chapter of NALMS, because 30 years ago, acid rain was a major problem in New England lakes. And so when we add sulfate to this process, here we have sulfate in the overlying water, shown here in the blue circle. And as it goes down into the water column, into the anoxic areas, it becomes reduced, and it forms hydrogen sulfide. Now, it turns out that the sulfur atom really likes to bind to iron. In fact, much more than phosphorus binds to iron. And so what happens, as these two combine and they form iron sulfide, it kicks off the phosphate ion, which then can go up into the water column and stimulate algal blooms. So if sulfate is a major source in your pore water, then you may have problems with respect to it binding to iron, and iron will no longer bind to the phosphorus and it can release the P into the water column. I want to talk about a few paradigms here. There's a general feeling that lakes that have extensive macrophyte beds are not going to be as problematic with internal phosphorus loading. But in fact, a recent study done in New Zealand showed that macrophyte beds were responsible for internal loading, because during the evening, you have a lot of respiration going on and the dissolved oxygen concentrations are driven down. During the day, when there's sufficient light, the photosynthesis drives the uptake of dissolved inorganic carbon, and the pH increases. And the combination of this results in a fair amount of sediment phosphorus release, in this case, largely from apatite, calcium-bound phosphorus. So again, if your lake, not that there's many, hard water lakes in New England, but if your lake is hard water and there's a lot of macrophytes present, it's entirely possible that they could be responsible for internal phosphorus loading. And then finally, we look at the physical and biological interactions. Again, this is what I've shown you earlier, the bioturbation from the carp or the gizzard shad and then the physical wind or boat turbulence that might resuspend these sediments that would go on, and then that would release phosphorus into the water column. Another paradigm is that polymictic lakes, lakes with short hydraulic residence times that would not be subject to stratification, will not have a large amount of redox-driven internal phosphorus loading. And I want to disabuse you of this because, in fact, these kinds of systems can have high internal phosphorus loading rates. Polymictic lakes can have low dissolved oxygen concentrations during calm periods or at night when respiration dominates. It's diel, it doesn't persist for a long period of time, but it lasts long enough, particularly in the early morning hours, late evening hours, to have internal phosphorus loading occur from redox-driven reactions. And then lakes with short hydraulic residence times also can go anoxic. And I'm going to use an example for the lake that my institute is located on, Muskegon Lake. And if you look at the hydraulic residence time, it's quite short, only 21 days. Take an aerial view of Muskegon Lake. The Muskegon River, the second longest river in Michigan, enters into the east end of the lake. It's about 7 miles out to this navigation channel where the water ends up exiting into Lake Michigan. We have a buoy in the middle of the lake, and we've been recording dissolved oxygen concentrations over the last 10 years. And here's the sensor for DO. Blue is 2 meters, green is 5 meters, red is 8 meters, and black is near the bottom at about 36 feet. And you can see in the summer months, in fact, the lake goes almost entirely anoxic for months at a time, which sets up perfect conditions for internal phosphorus loading despite the very quick hydraulic residence time in these lakes. Moving on to the third area is how do we measure internal phosphorus loading? There's a lot of different ways to do this. I just want to present 3 of the more common ones. This includes measuring the total phosphorus in the sediment, then using sediment core incubations to look at the gross benthic flux out of those sediment into the water column, and then finally going out into the field and measuring the accumulation of P in the Hypolimnion. So Gertrude Nurnberg did a study back in the late 80s where she looked at 7 lakes, dimictic lakes from Ontario and Connecticut, and what she found was a very strong relationship between the total phosphorus in the sediment and the internal phosphorus loading. It was higher for the wet sediment than the dry sediment, but the R-squared values were still pretty good. However, when she then looked at the fraction of the total phosphorus in the sediment that was mobile, she looked at the bicarbonate buffer dithionate, which is the redox-sensitive hydroxides of iron and manganese. She found the relationship was even stronger with these fractions. And what this goes to show is that measuring total phosphorus can give you a misunderstanding of how much phosphorus is available to be released from the sediment, because total phosphorus can combine both mobile fractions and stable fractions, and the stable fraction isn't going to go anywhere. And so what you really need to do to use sediment phosphorus is you need to fractionate the sediment to see what form the phosphorus is in. And there's a lot of different extraction procedures that are out there. I'm using one here from Hooper from our book, and I'm not going to go through all of this. You can look this up on your own, but here we see the 3 forms that are mobile, and then 4 additional forms that are stable. And if it turns out that most of your total phosphorus is in the stable form, it's going to grossly overestimate the amount of phosphorus that could diffuse out of your sediment as part of internal phosphorus loading. So strongly recommend that if you use total phosphorus as an estimate of what your internal phosphorus loading potential is in your sediment, that you fractionate them as well. For sediment core incubations, here's a cartoon that shows inside the core tube, you have your sediment, you have your water column. You can add some kind of chemical inactivant to do experiments if you want. This shows you the core tubes in our lab in a growth chamber. In this case, we're adding either air to be oxygenated or nitrogen to be anoxic. And then we can also control for temperature. We can replicate, so it allows a lot of manipulation to occur. That's the real advantage of this. And if you're interested in looking how to do this, rather than me trying to explain in this webinar, I would suggest that you take a look at this URL. You'll be able to see this afterwards when Amy puts this up and posts this. But this is in the Journal of Visual Experiments, and it basically is a video that shows exactly how we do this and how we calculate internal loading. And it gives you a very good idea of how to do this process. The third way is to look at phosphorus accumulation in the Hypolimnion. And in this case, what you do is you break your lake down into different layers to measure the volume of each of these layers. And of course, not all lakes are going to be this regularly shaped, but by basically integrating over these different layers, you can look at the phosphorus in each one of these layers. And what Michael Hupfer is suggesting is that you then look at the phosphorus in each one of these layers over time from the point of stratification, either to the maximum point of phosphorus release or over the entire period of stratification to figure out how much of your phosphorus is in each of these layers. You then sum that up over your entire time period to see what that total phosphorus release is in your hypolimnion. And if we just do a review of these three different methods, if you look at the phosphorus in the sediment, it's quick, that's the advantage. And if you're looking to where to apply your chemicals, if you don't want to apply chemical inactivants over the whole lake, it can tell you where the hot spots are and the hot moments in your system. But of course, we know that sediments are spatially heterogeneous in lakes. And so because you're only sampling a small area, you're not going to be able to account for the heterogeneity. And it's a very coarse estimate of internal phosphorus loading, which is why it's important to do the fractionation. For the incubations, they're controlled, so you can manipulate variables, you can get replication, that's all advantageous. But again, your sediments are spatially heterogeneous, and taking these small sediment cores, there's no way to cover the total heterogeneity that goes on. For the hypolimnetic phosphorus accumulation, it removes that heterogeneity, that's a major advantage, but it's time consuming, and depending on how many layers you have and how often you sample, it can become quite expensive. And then finally, with regard to the relative importance of internal phosphorus loading, I caution people when they do this kind of analysis that it's really arbitrary. It really depends on how you're calculating your internal and external loads. For internal, are you accounting both for oxic and anoxic release? Are you looking at the seasonality? Because the release rates are generally much higher in the summer than in the winter, because those warm temperatures stimulate the bacterial mineralization of the organic matter. For the external load from your watershed, are you looking at both storm flow and base flow? Are you including all the external sources, including things like atmospheric deposition, to really know how much phosphorus is coming in from the outside? So it's really possible, and you see this, that people cherry pick the data to show high or low relative importance of internal phosphorus loading, depending on what their ultimate question is to be answered. And here's one example from a study by Song et al. In this case, we're looking at the annual external load in gray relative to the internal load in black. And then on an annual basis, you can see that in all four lakes, internal loading is a very small percentage of the total external load. But now, if we move to the summer, we see that, particularly in Bluestem and Conestoga, the internal load is accounting for well over 70% in these lakes. So it just gives you an idea how different these relationships can be depending upon how you look at your data. And the same thing goes for measuring release rates. And these are from data that we published almost 15 years ago, or more than 15 years ago, where if we start off at the 1 day and then we measure it up to 12 days over this linear release rate, we can get a fairly high release rate of phosphorus from the sediment. But if we go all the way out to 20 days, we see now this release rate is about half of looking at just 12 days. So again, depending on how you present your data, how you calculate your data, you can show high or more moderate release rates. So you need to be very careful about how these release rates are being calculated. So recommendations, if you're looking at relative importance for internal phosphorus loading, strongly recommend that you compare across consistent time periods, either an entire year or just the summer months. Make sure you're not comparing apples and oranges. Look at the variability. Don't report just one data set. Look at a series of data sets so people understand that there is natural variation going on in these kinds of measurements. And then if at all possible, do this over multiple years as longitudinal studies, because we all know that some years are going to be biased to be dry or wet, and they can skew our results. So where are our knowledge gaps and what do we need to do? One, we need standard methodology for measuring internal phosphorus loading. If you look around the world, people are using all sorts of different measurements, and it makes it very difficult to assess commonalities and figure out what the best management strategies are. We also need to understand the drivers of this variation across different scales. Looking at seasonality, the different geochemistry of the sediments, the hydrologic connectivity within lakes, all of this influences the internal phosphorus loading, and we simply don't have a good handle on a lot of this. Legacy phosphorus in our watersheds is another unknown. We know it's important, but often we don't know where it is, how much is being released under different circumstances, different time frames. All of this needs a better understanding. And then finally, we need to look at this from a multidimensional perspective. We're focused on the technical results because we know what the technical answers are, but we also need to think of this in terms of the socioeconomic aspects as well to get a better understanding of this and understand how society reacts to these kinds of issues. So with that, I want to thank you for your attention. I also want to pay homage to my alma mater, UVM, is where I got my undergraduate degree, to show that I do have some New England credibility in this whole process. Thanks, Amy.
Thanks very much, Dr. Steinman, for your presentation. So if you could click out, perfect. And then I'm just going to switch over to Dr. Ken Wagner for his presentation.
We looking good here?
Looks good, Ken. Thanks.
Is everything visible? Very good. Okay, so Alan's given you a very, I want to say thorough because I've read the book and there's a lot more to it, but a solid explanation for how phosphorus winds up being recycled internally loaded in a lake. The next link in this is how this figures into cyanobacteria blooms. Certainly the phosphorus figures into any algae bloom, but there seems to be a particularly strong link to cyanobacteria. Okay, first, just so we're all on the same page, what are cyanobacteria? Hopefully most folks on this webinar kind of know this, but just to make sure we're all together, these are photosynthetic bacteria. It's a very old group. They've been around long before we were. It's a very diverse group. It is not true that all cyanobacteria can be lumped together for purposes of characterizations any more than people can be. They are native, they are natural, they are part of a functioning aquatic system. Certainly the hot spring algae in Yellowstone are mostly blue-greens. Some of the cleanest lakes I've worked on have benthic blue-greens. It's fairly common. They're not all bad. They are mostly small cells in large aggregations. So you have tiny cells, a few microns across, but big particles, either chains of beads like here with the Dolichospermum on the left, or Microcystis on the right, where you've got these tiny cells. They have preferences. Again, there's a lot of variability, but they like warmer water. That's a simple matter of they largely metabolize sugars, which are best metabolized at warmer temperatures. That does not mean they can't occur at cold temperatures. I've seen blooms under the ice, but they prefer it. They like elevated phosphorus. They're actually not the best competitors for phosphorus against some of the other algae, so they do very well when there's plenty of it available. They like reduced iron, which of course tends to come out with the phosphorus in that chemically induced internal recycling, because the iron figures into nitrogen gas fixation, which is something specific to them. I'll come back to it. And they like higher pH. The higher pH pushes you from carbon dioxide to bicarbonate. Blue-green algae are better at using bicarbonate than a lot of other algae. So there's ecological reasons for all this. Pigments allow photosynthesis under low light. Blue-green algae have chlorophyll A, as do virtually all of their algae, but it's not the dominant pigment. They have a lot of other pigments, which is where the blue-green color and a lot of other colors come from. And those allow photosynthesis under fairly low light so they can grow deeper. Many of them have these heterocytes, which are specialized cells where gaseous nitrogen can be fixed and turned into the building blocks for proteins. Other algae can't do this. They need nitrate or ammonia or some other form in the water column. The blue-greens, not all of them, but the ones that have the heterocytes can get away without it and use the raw nitrogen gas. Many of them control their buoyancy. That is a huge factor. They can create gas vacuoles, and here it's planktothrix on the left. All those little white dots are gas pockets in the cells, and those will allow them to float, and by controlling the number and size of those gas pockets, they can actually control their buoyancy. It's pretty sophisticated. They're not terribly good at it, and they wind up forming surface scums and getting fried in the sunlight a lot of the time, but they can do it. Resting stages, which is true for most algae, tend to fall sediment and germinate later. This happens to be Aphanizomenon on the right, and it has these light whitish sort of or cream colored spots are the formation of their resting stages. that then drop to the bottom and can come back later, germinating, growing down there, and then popping up. Most cyanobacteria, synonymous for blue-green algae, and don't get hung up with that. I've heard people say there's no such thing as harmful algae, it's only harmful cyanobacteria. Cyanobacteria get lumped in with other algae. It is true that because they're prokaryotic organisms, they are fundamentally somewhat different, but the term algae is just a common name for all these organisms. Some of them could be considered protozoans or other things. When you get to that level, there's a lot of overwash between the groups. Point is that most cyanobacteria are capable of producing toxins. We used to say it was very specific ones. The more we do, the more we're finding that they almost all have some ability to do this. Whether or not they actually make the toxins is another story. If you look at a lake and it's just green with blue-green algae and you just know this is a messy spot, you still only have about a 1 in 5 chance of there being toxin at a dangerous level. Now, 20% is not something to mess around with, but the bottom line is just because it's got blue-greens in it does not mean there's toxins present. Again, it's a diverse group, extensive taxonomic revision in the last decade or two, which is a real problem for terminology. Just by way of some of the ones that are out there, Aphanizomenon, which I've already mentioned and showed, they split a group off of that and they call it Cuspidothrix. Those are ones with real attenuate ends. Dolichospermum, which is actually what most of what we knew as Anabaena became. But Anabaena still exists as benthic forms that don't produce those gas pockets and don't float. So the ones we're really concerned about really are the Dolichospermums now. And there's a few other ones that have been broken off from that. Microcystis. Fortunately, Microcystis stayed Microcystis for quite a while. Back in the 60s, somebody changed it to Anacystis and lumped some things into it, but Microcystis is still pretty solid. Although we found that the separation of species based on physical features turns out not to be very good. Warnicinia, which used to be called Coelospherium, but there still are Coelospheriums out there. And several others, Snowella and other things have been broken off from it. So these are, there's a lot of variability here, and there are ecological factors that figure into this. So there's a lot to know in terms of blue-green taxonomy. It's just like if you had a plant problem and you went to somebody and said, I've got a plant problem, they'd ask you what the plant is. Well, if you have a blue-green problem, we're going to ask you what the blue-green is too. So you need that analysis to know what you've got. A few other ones, Planktolyngbia, which we used to call Lyngbia. There still are Lyngbia, but this one's separate. These are ones that float. Planktothrix, which I mentioned before, split off from the Oscillatoria group. Often toxic, worries me a lot, forms metalimnetic blooms. It's been a source of problems in a number of New England lakes. Raphidiopsis, which used to be called Cylindrospermopsis, which used to be called Raphidiopsis, it's going back and forth, is more of a tropical one, but we find it in the north. I found it in Massachusetts. I know it's been dominant in a few Indiana lakes, so it can be here. And then Gliotrichia, which is one I mentioned because this is kind of the one that kicked off the whole idea of cyanobacteria and internal loading being linked. Kaylin Carey, when she was an undergraduate, she's now a professor at Virginia Tech, and a number of colleagues from various New England universities all got on this and found that this fuzzball basically grew to that size at the sediment water interface, and then at the very edges of each of those filaments, these long wispy things you can't really even see here, which are gas-filled cells which allow the entire colony to come up and float, bringing with it all the nutrients that's accumulated from down at the sediment water interface. So it's actually not a symptom of eutrophication. It may actually be a cause of it by bringing those nutrients up. Alan talked about this as one of the modes for internal phosphorus loading. Okay, so modes of bloom formation. There's basically 4 ways you get a cyanobacteria bloom. Entry from upstream source in large quantity. Yeah, okay. So there's a lake upstream, it flows into your lake, and that lake had a lot of blue-greens. We're not really talking about that today. Organic growth in the upper water layer. This is how we used to always think it happened. You had a few cells, there was too many nutrients, and they grew out of control. Turns out that's not the common means by which we get blue-green blooms. Number 3 is the formation in a mid-depth with movement into the upper water layer, basically what we call a metalimnetic bloom. It forms down around the thermocline, usually slightly below it, where there's just enough light to get by, but there's enough nutrients coming from underneath to fuel the bloom. And then depending upon where the thermocline is, those things may get mixed into the upper water column and cause a bloom. And then finally, the one that's turning out to be way more common than we ever thought is this benthic growth of planktonic forms, followed by a synchronized rise into the upper water layer. And some of the ones that are involved, Raphidiopsis and Merismapedia, are ones that do actually tend to grow organically in the water column. Planktothrix and Pseudanabena are very commonly metalimnetic bloom formers, and Microcystis, Dolichospermum, and Gliatrichia, ones that are all associated with potential toxins and are often bad bloom problem species, tend to form as this benthic starter growth that then floats up. So the factors that support the cyanoblooms, the increased temperature, the warmer it is, the more they like it, and you get faster growth rates, and they are metabolically favored. You can know a lot about succession of algae just by understanding their food resources, how they store food, and what the temperature is. It'll tell you a lot. Increased nutrient inputs. Well, of course, that's important for all algae, but internal recycling, internal loading of phosphorus, those legacy inputs, can be a main source of phosphorus, particularly in the summer when it's warmer, just like Alan showed. And those have a tendency to come with a low nitrogen phosphorus ratio. Nitrogen's also getting released from the sediments, but not nearly as high a level as the phosphorus, and you wind up with an excess of phosphorus versus nitrogen, and that favors those blue-greens that can use the nitrogenous gas and love having that extra phosphorus. All right, this is sort of the crux of this presentation. Here's a sequence of oxygen profiles in a theoretical North Temperate Lake. In June 1st, we've got a low oxygen right at the bottom, and what shows here is about 12 meters, and it tails off or goes up pretty quickly, and in the upper waters, there's plenty of oxygen. As the water warms, it holds less oxygen, so the upper waters have less oxygen over the course of the summer. But more importantly is this interface of low oxygen water gets higher and higher in the water column. So more and more area, more and more volume are subject to this low oxygen. Now, if we look at a cross section of the lake, that oxygen here in the orange corresponds to June 1st in my theoretical diagram. There's what's contributing. It's not that much. I wouldn't say it's nothing. It might be important in the long run for the lake, but it's a long run up to the thermocline or the area below it where there's enough light for algae to grow. It's a small area going into a big volume. It may never figure into blooms. But by July 1, now we have a bigger area exposed to low oxygen. And by August, an even bigger area. If we extended it out to September, we might actually have it all the way to the thermocline where this entire area is anoxic. It's all contributing phosphorus by that chemical redox mechanism that Alan spoke of. And now we have the ability to support metalimnetic blooms, or just to have enough phosphorus leaking across the thermocline to support the organic growth in the upper water level. The depth at which the thermocline forms is really important. And that's a function of the spring weather pattern. And of course, lately, try to predict that. The cyanobacteria, in a particular lake on Cape Cod, these are real data. You had the thermocline down around 6-7 meters, and slightly below it at 8, we had a very thick bloom of planktothrix, and it was toxic. And that was in 2008, but it never came up. It never got mixed. The thermocline goes down as the fall turnover begins, and this stuff never really showed up in the upper water column. The very next year, 2009, the thermocline set up much shallower. The thick layer was at 5 meters, and an August windstorm mixed that, and we had a tremendous toxicity event. A lot of mussels died. So this is something that you can't predict from year to year, at least not well in advance, and it makes a lot of difference to what's going to happen. All right, so those cyanobacteria blooms with a benthic origin, which I'm saying are way more common than we used to think. If you look at a typical deep lake, you've got this whole area down here at the bottom contributing phosphorus because the sediment's anoxic, but there's insufficient light to grow anything. So below that green line, that's the compensation depth, there's phosphorus accumulating. It may eventually cause a problem in the upper water, but at the moment, it's not really going to grow a lot of algae. But as that anoxic zone creeps up in the water column and we get up in the zone around the thermocline where there's enough light for these blue-greens to grow, they can actually grow right at the sediment-water interface. The phosphorus never has to make it into the water column. They can pick it up right at the surface, grow, and then pop up to form the bloom. So you actually get a donut around a deep lake that's contributing. This happens a lot with Gliotrichia. It also happens with Lichospermum and Microcystis. So it's a real issue in that we have this intermediate depth zone that contributes these algae. Now, if you move over to a shallow lake, and again, Alan's explained this very well, the assumption's always been that, well, you don't get all that much phosphorus making it to the upper water column because we have this process where if the ferrous iron, which is the soluble part with the phosphorus, is released into the overlying water column, there's enough oxygen around, it will recombine, become ferric iron with phosphates, and drop back out. This is tongue-in-cheek called the Ferris wheel. Cute. You know, it goes around in circles. And the point is it's not up here long enough to make a great deal of difference to the water column. But again, think back to the last slide. Instead of a donut of contributing area, now we have this anoxic sediment all the way across the lake, the entire shallow lake, except maybe for the very peripheral areas that are sand, gravel, rock, or have plenty of oxygen. This can go anoxic, or at least low oxygen. You can get that release of phosphorus from redox reactions, and the algae are right there to pick it up and run with it. And then they'll pop up, and all of a sudden you have a bloom. If your lake is one that seems to get a bloom almost overnight, short of a week, it's almost undoubtedly happening this way. All right, so why are cyanobacteria more linked to internal loading than other algae? Well, it's true. Other algae can utilize the same approach. You think about big filamentous green algae mats. They do the exact same thing. They grow at the sediment-water interface. They trap their own photosynthetic gases, and they eventually float up. Golden algae will make metabolism blooms that can be a real problem for water suppliers because they're often taste and odor producers. In the south, we have a couple, Primnesium, for example, that can be toxic. But really, the issue here is just that they're making use of this low light but adequate nutrients in the Hypolimnion. But the cyanobacteria are better adapted to low light. They can grow deeper. They have the buoyancy mechanism to move up and down in the water column. And they like that internal nutrient release because it has the low NP ratio and a higher ferrous iron content that favors those cyanobacteria that can fix the nitrogen gas. So a word about temperature, oxygen demand, and internal loading. This is sort of the climate change part. Hypolimnetic oxygen demand in grams per square meter per day is a big factor in whether or not the bottom of the lake goes anoxic. You'd like to be down around 0.5 grams per square meter per day or below it. That's considered an oligotrophic lake in most cases. This lake I was working with for the last decade, actually the best they do is around 1 gram per square meter per day, which means it's going to be anoxic by the end of the summer. That's enough to do it. But when the water temperature is warmer in the bottom, just by virtue of the way things set up in the spring, it can literally double the oxygen demand. And now this is going to be going anoxic by the end of June. Well, that's a whole lot of difference in terms of a month or so of internal release versus this huge release going on for 2 or 3 months of time. So there's a lot of variability, and that's another one. If the phosphorus levels in your lake vary tremendously from year to year, you have to look at the external loading and see if it corresponds to precipitation and land-related factors. But you also have to think in terms of how is the lake different this year than last year in terms of its temperature, the area exposed to low oxygen, and the potential for internal release, because that variability can be very high. All right, cyanobacteria bloom trends, they are increasing. We've got articles in Lake and Reservoir Management going back over a decade now that document this. People used to ask me, is it really that these blooms are increasing or are we just more aware of it? And the answer is yes. It's both. We're studying it better, but yes, it is more frequent. Phosphorus has been rising. We're not doing a good job controlling it. And we get this decreased nitrogen phosphorus ratio, which favors the cyanobacteria. Temperatures have been rising. Again, this isn't a climate change talk, but there's no question about it. The temperature issue is a big one. We're getting more oxygen demand, more internal loading, plus warmer water. It all favors the cyanobacteria. And those bottom growth, quick rise blooms are increasing in particular. We've got a long-term accumulation of nutrients in the sediment. It fosters those growth. When you hit this tipping point of low oxygen over a bigger area, it's a huge influence. The light and the low oxygen in sediment are key triggers. The intermediate depth zone is implicated as the biggest contributor in deep lakes. In a shallow lake, it could be the entire bottom of the lake. Just a few references, and this will all be available to you online. There's lots out there. I picked out these 4 because I thought they pretty well summarized what I'm saying, and they're relatively recent. I don't have any connection. I know some of the authors. None of this is my work. None of it's published in Lake and Reservoir Management. This is an honest appraisal of 4 references. If you want to understand this factor, this is a great place to start. And I will stop there. We're not really going to do questions and comments for individual speakers. We'll come back to it at the end, but if you've got them, by all means, type them into the chat box and we'll deal with them. That'll do it, Amy. And I can stop showing the screen.
Great. Thanks very much, Dr. Wagner. And now we are going to move on to our final presentation of the 3 with Dr. Michael Lurling. And he's going to talk with us about how to control some of this internal load.
All righty. There we go. Right. So, are you able to see my screen?
Yes, we have you. Thank you.
Oh, fantastic, fantastic. Thanks a lot for inviting me to this nice webinar. And thanks a lot, Al, for introducing internal phosphorus load, and Ken for highlighting the link with Cyanobacteria, which nicely paves the road to my presentation, what to do about it. And I often get this question. We do have a eutrophication issue in my lake. What to do about it? Well, always start with a proper diagnosis. Start with a system analysis, because each lake is unique. And a couple of years ago, there was this nice item in National Geographic written by Lisa Boyer, and I was intrigued by these satellite images that came along with it. And if you look at these images, you can clearly see that all these bluish or purplish dots or shapes, well, they have different shapes. These are different lakes, different colors. So already emphasizing that these lakes are different. So please start with a proper diagnosis. Start to figure out where the water is coming from, where the nutrients are coming from, and where your cyanobacteria, if they are causing the problem, are coming from. Of course, it's always good to start with controlling external load. You will never get regret from that. And the magnitude or the extent in which you are able to reduce that will also determine how effective additional in-lake measures might be in speeding up recovery. We'll base this presentation primarily on commonly used methods that can be found in this book that Alan and Brian Spears edited, that appeared earlier this year, and these methods are summarized in chapter 5 of that. You can easily or try to remove nutrient-loaded sediments, try to extract nutrient-enriched hypolimnetic water, or try to cover sediments with sand or clay. Implement aeration to promote this iron trap that Alan explained, or use other amendments to keep phosphate stored in the sediments. We all know that nutrients may enter our lake via numerous sources. And so they can come through pipes from industries or from municipalities or wastewater treatment plants. And those are, in theory, easy to tackle. But even in high-income countries, only about 70% of these point source nutrient pollution is being treated adequately. In low-income countries, it's less than 10%, so still a lot of needs to be done there. Unfortunately, we also have all kind of diffuse sources, runoff from agricultural fields, urban runoff. As Alan mentioned, atmospheric deposition should not be neglected. And related to the rainfall runoff cycle, we also have leakage of nutrients from fertilized or over-fertilized soils into groundwater, leakage from septic tanks, etc. That will eventually fuel our lakes with nutrients via nutrient-rich groundwater. And in my country, this is really a big issue. We have a massive agricultural industry, and several drinking water wells had to be closed due to, in this case, huge amounts of nitrates accumulating in the groundwater, far above 50 milligrams per liter. This ongoing pollution, of course, will build up phosphorus pools in lake beds, and that's what's causing our internal load. This point source reduction, it's always a good thing. So if you can do it, please do it. And depending on how long the lake has been loaded or how severe your internal load is, recovery can be very rapidly. And there's a nice example from Lake Washington where after diversion of external load, the lake recovered within more or less 5 years' time. And this can be seen in this graph here. Total phosphorus, indicated in black squares, drops rather rapidly after the input was diverted, and also chlorophyll concentration dropped rather rapidly. Might take a bit longer in other lakes. So this lake in the UK, it took about 20 to 30 years before it returned to pristine conditions, but recovery may happen rather rapidly. But this is unfortunately not always the case. We have examples from Denmark, for instance, Lake Muskegon, that is still in a bad ecological condition, despite all kinds of measures have been implemented already decades ago. Started in 1976 with the sewage treatment plant, improved in 1982 with removal of phosphorus. There was even a slaughterhouse and it still discharged something that was diverted, but still nutrients and total phosphorus concentration remained high. In the diagram here, the symbols are the measurements and the line indicates the modeling exercises. So even after 30 years, the lake is still suffering from high phosphorus concentrations and from cyanobacterial blooms. And the researchers involved in this case, they concluded that internal phosphorus load is an absolute necessity because the lake doesn't show any signs for further recovery. So they need to do something more over there. Why does it take time? Well, it has to do with this internal storage. If it's large and if you cannot remove a huge amount of it within a few years, it simply will take time. So if we have a large input and we have some outflow and we have some sedimentation and then building up of the internal load, it really depends on how much can be replaced with lower phosphate or lower phosphorus containing water. So the residence time, but also the amount of nutrients that can be removed, it really determines how long it will take. Of course, this diagram here, water depth over time and an accumulation of phosphorus in hypolimnetic region, as has been mentioned in the previous presentation, that can be used to speed up recovery. And one of the things that has been done quite effectively is extracting that water, so hypolimnetic withdrawal. Gertrud Nürnberg, as she was already mentioned before, she wrote 2 very nice reviews about this intervention method. And the last one was published last year in 2019, where she summarizes all results that have been obtained with this extraction using an Olszewski tube and extracting this nutrient-rich water and discharging it downstream. And what she found is that the more water you can extract, the stronger the response will be. So the more nutrient-rich water you're able to extract, the faster recovery will occur. Of course, this water is not only rich in nutrients but also in reduced compounds. And so Alan also mentioned that it might be full of hydrogen sulfides, or might also contain manganese. So it's better not to discharge this directly in a downstream lake or water body, but to do some treatment before discharging this water to prevent negative side effects in receiving waters. Another aspect and the other side of this, where hypolimnetic withdrawal makes use of the anoxia in Hypolimnion, and wants to have as much as phosphate being released from sediments, aeration and oxygenation tries to reverse that and try to keep as much of the phosphate bound to iron as possible. And as Alan already explained elegantly, the iron, phosphorus and sulfur ratio is very important there. I will not go further into detail. He has explained that already very elegantly. And there are numerous well-studied systems already running with quite high efficiency for several decades, and a few reviews have been published about them. Nowadays, most of those oxygenation or aeration devices are aerating the Hypolimnion and are designed in such a way that they maintain stratification, and therewith also maintain a cold water habitat for fish, and you're not warming the entire water column and also warming the sediment that may stimulate all kind of organic matter breakdown processes. So there are a lot of different systems available now. In this screen, we have partial lift or full lift, or diffusers or species cones. One of the things that I should also mention are side stream supersaturation units, in which hypolimnetic water is being pumped up. It's supersaturated with oxygen and then pumped back into a diffuser. So maintaining stratification and enriching the oxygen concentrations in the sediment. Well, as a rough estimate, it's best to keep oxygen concentrations above 5 milligrams per liter in the Hypolimnion to ensure that you have enough oxygen on top of the sediments that allows your sediment trap, iron trap, to be effective. There are some other new techniques. I'm not aware of any lake trials with nanobubbles. Looking at the devices, they might work in small ponds or maybe small lakes, but I'm not sure if they are powerful enough to do something in big lakes. We'll give you an example of hypolimnetic oxygenation from the Netherlands. It's Lake Oudekerkerplas. It's a 0.7 square kilometers lake in the south part of the city of Amsterdam. It's sand excavation. The sand was used for the motorways here and for construction works in the city. And Vattenfall, which is an electrical power company, is using the hypolimnetic water to cool offices in summer. And by doing so, they are able to reduce the carbon dioxide emissions quite substantially for that region. Since they're using hypolimnetic water, they are not allowed to discharge the warmer water in the Hypolimnion because it would fuel cyanobacterial blooms. So they implemented a pure oxygen injection system near the bottom to precipitate the phosphate with iron and then discharge phosphate poor water into the Hypolimnion. And some of the results that was given presented to me by provided to me by von Smollers. This total phosphorus concentration in the Hypolimnion in red dots are this is the cause of total phosphorus during gradual eutrophication of the lake. And then when the system starts to operate, the total phosphorus in the upper water lake started to decline. There was a malfunction of the system in the beginning, so it went up the next year. So it really underpins the necessity of keeping running these devices. If you don't do that, you will be back to business as you had before, the situation as you had before. But it really seems to work fine in this lake. Then another very widely applied intervention, at least in my country, sediment removal. We have no idea how much this has been done. We know that a lot of authorities are doing dredging and excavation on a regular basis. This is part of their regular maintenance interventions, not only for nautical reasons, but also for improving water quality. It has, by definition, a strong impact on the aquatic system. If you remove all the water, or mostly of all the water, well, of course, and then excavate, that has an impact. But also the dredging, the stirring up this anoxic sediment will lead to, even if done in this case in later winter, early spring, results in low oxygen concentrations and highly turbid water, even fish kills. And this guy, I hope you can see that he was trying to rescue some of the suffocating fish. He even got fined by the police because he had no permit for fishing. But that's typical Dutch, of course. It comes with quite some costs, these interventions. A rough estimate is that it costs about 50 euros per cubic meter in my country to dredge and to also to deposit the sediments. An alternative is not to remove the sediment, but to cap it with a layer of sand or clay to form a physical barrier, trying to prevent diffusion of phosphate to the overstanding water. Of course, that's what we call a passive cap. It's also possible to mix this material with some compound that has strong phosphorus binding capacity creating a more active barrier. This is explained in the book on lake restoration, published last year, and this is chapter 7 by Max Gibbs and Chris Hickey from New Zealand. The final category is using products to keep the phosphorus in the sediments, or to strip a water column and then keep it in the sediments. Aluminium salts are widely used. I know that alum is one of the most favorite, I think, most widely applied phosphate binders and coagulants in the US. Polyaluminium chloride is more applied in Europe. Iron salts can be used, calcium salts, but only at relatively high pH, because otherwise your products might dissolute again. And modified clays, such as phosphorus and lanthanum, modified bentonite, and which is widely used nowadays throughout the world. More than 200 lakes have been treated with that. Aqua P is a novel product, which is aluminium modified zeolite from New Zealand. And I'm not aware of large all lake applications, except some trials in New Zealand, but it's being studied now. As Alan also mentioned, sulfate is a tricky substance. I will not get a permit in my country to propose, or we will not get a permit to use alum or iron sulfates because of the sulfates and because of the risk of stimulating internal eutrophication. So that's a no-go for this country. I have asked Dr. Ezekiel Oziri for this nice picture of some treatments in sediment cores, because he included something it's not being done that often anymore. It's a combination of iron and nitrate. Nitrate has an additional electron acceptor. And this is in former days was called a Riplux treatment, but from the cores it looks nice, and over time as well, we can clearly see that iron remains oxidized, and also the top layer of the sediment is oxidized. And I know from his data that it really lowered and strongly inhibited the phosphate efflux from the sediment. And then the funny thing is, he told me that these layers were based on removing the same amount of phosphorus. You needed more iron nitrate than you needed phosphorus, and you had a fluffy layer of aluminium flux that gradually are being compressed. Those experiments are very nice and important to test the efficiency of all kind of different products. I'm giving here an example with sediment cores from an extremely diluted urban lake under oxic and anoxic conditions and controls. Phosphorus is lanthanum modified bentonite, so bentonite is just the control for the physical capping that the phosphorus layer would do. And zeolite, that's the control for the Aqua P, which is a modified zeolite. Both were able to reduce phosphate release strongly and under oxic, but also under anoxic conditions. So I will show you a case study from the Netherlands, Lake Houbraken. It's a small lake, it's a sand excavation dug in 1967. It gradually became eutrophied and was closed for 4 months in 2007. It has no direct inflow, there's no stream coming in or going out. It's fed by groundwater and precipitation, and measuring the water fluxes, but also measuring the amount of phosphorus in there, made clear that sediment phosphorus release was really the main source of phosphorus in this lake, and was fueling the blooms. It's the black bar in the diagram over here. So we came up with an intervention first to clear the water column of all particles using a coagulant, polyaluminium chloride, slightly buffered, and using some phosphorus as ballast to get enough waste into the flux. So stripping the entire water column and then adding some phosphorus to have it settle through the water column and create an active barrier on the sediment. Well, if you strip a water column of all particles and then add a clay, you create a glacial lake. So it looked a little bit bluish after the intervention, but when that was settled down, the water was pretty clear, and we had more than 10 meters transparency, which is rather unique or maybe ridiculous for the Netherlands. Going to some of the results, on the y-axis is the water depth. It's on the fixed station in the lake over time. This is the moment of intervention. Before intervention, we had relatively high total phosphorus concentrations on average, but this is the median 134 micrograms per liter. And after intervention, it was down to 14. And over time, it becomes more greenish and yellowish. The total phosphorus concentrations are increasing again. So the lake will return to a more eutrophic state. And this has to, and this is due to ongoing external load. It's impossible to cut off the groundwater, or it's an official bathing site, so you will not stop civilians to have a swim in there. It's quite difficult to reduce leaf litter or to scare off the water birds. So these sources remain, and they are building up an internal load again. We're pretty sure this has to do with the external load and not by the product used losing its binding capacity, because we have sediment cores that are in our lab already for many years now, and we still have a very low phosphate concentration and have no signs of internal load again. So this is external load building up a legacy again. For this lake, the application costs us 50,000 euros with a cost of 150,000 euros. So it's a few thousand euros per season. A repeated intervention would be very wise, and you could see this as kind of maintenance of the water. It's much less than the money that's being spent to maintain the beaches and the lawns in the recreational area. A final example is about multiple measures. There's another urban water body in the southern part of the country, also relatively small, 4.8 hectares, 1.2 meters, very shallow, but had 2 feet mud layer on top of some peat. There were no macrophytes in there. Well, from the green spot, you can see that there were quite some cyanobacteria and almost 270 kilograms per hectare of fish, which is not even that bad, because in several of those water bodies you can easily have 1,000 or 1,500 kilograms per hectare, dominated by carp, which is also fairly normal in our waters. The system analysis gave us that internal loading in black in the dark was a problem, but also the external inflow via 2 small streams. So if we wanted to restore this lake, we needed to tackle both to come at least in the region of those critical phosphorus loads that we determined with the model PC lake. So a whole package of measures was needed to restore this lake, and first of all, fish was removed. Then the external load, the streams were diverted, the entire water body was dredged, and the banks were reconstructed. But because the mud was laying on the peat, and we don't want peat to oxidize and to release nutrients, we had to cap that. So we put a layer of 30 centimeters sand on it, which was enriched with some phosphorus, creating a more active barrier. Adding sand on peat and doing dredging activities, you have nutrients in your water column. So we also treated the lake with a little bit of polyaluminium chloride and some additional phosphorus, and also started to plant Macrophytes. Well, these are the results of total phosphorus 3 years before the intervention, the year of intervention. And you can see before nice seasonality. So these peaks in summer clearly indicates the internal load. So this is a very shallow water body, 1.2 meters. So it's another example that also in very shallow waters, you can easily have internal load issues. And after the intervention, total phosphorus remained very low, and it still is. So I haven't added the data of 2009, 2020 yet. And the blue line indicates the target that was desired by the water authority. Also, cyanobacteria chlorophyll concentrations remained fairly low after the intervention, so everybody's happy there, except the fishermen. They keep on stocking the water with carp. So I think the water authority already had to remove carp 6 or 7 times now, but that's well, we have to deal with that. That's funny. To summarize this, if you want to do something about internal load, please always start with a diagnosis, with a system analysis. Try to find out where your water and where your nutrients come from. And external load control, that's very important. If you're able to reduce that, it really will have an effect on the longevity of interventions you will do in your lake. Your diagnosis will also give you underpinning if it becomes too costly to do something. So it also will give you, well, at least some arguments to say, well, in this lake, we're not going to do anything. So unfortunately, in many places nowadays, repeated interventions are unavoidable because we have these ongoing diffuse loads. In my country, even if we would stop with all agricultural activities, it will take, well, more than a century before all nutrients that are stored in the over-saturated soils will be leached out. So we will keep on fueling our lakes. What to do? Well, that depends on your system analysis and depends on the characteristics of your lake. You can do hypolimnetic withdrawal or oxygenation or dredging or chemical phosphorus fixation. They can all be used. I want to mention that after 14 years of research on Phoslock, that is indeed one of the most, I think, the most powerful phosphate fixative we have nowadays in the market. It's effective in a broad pH range. It works on anoxia, and it forms a stable mineral rhabdophane lanthanum phosphate. So phosphate will remain permanently bound to the lanthanum. It's no longer available to biota. So that's a good thing and a nice tool to use. It was my pleasure to present briefly some of those techniques that are out there. I hope, and I'm sure that quite a few of you online are doing a lot of great works on lake restoration. Please share your adventures also online, which is even more important nowadays, so we can also learn from that, and so that we do not reinvent the wheel, or maybe have some other great innovative techniques that might be useful for us here in Europe as well. So thank you very much. Thank you, Amy.
Thank you, Dr. Lurling, and thank you to each of our presenters in this session. We have had a number of questions come in during the presentations, and I'm going to go through and pose those questions to each of you. I will try to sort them and assign them to you as they came in when you were presenting. So the first question is probably for Al, and it's from Tim Sorrentino. And he asks, would mechanized lakebed aerators cause sedimentary phosphorus to be released into the water column?
I'm sorry, Amy, can you repeat that question?
Sure. Would mechanized lakebed aerators cause sedimentary phosphorus to be released into the water column?
Well, in theory, the aeration should prevent it, but I guess there's the possibility, depending on how the aerators are designed and implemented, that they could resuspend the sediment in that process, particularly if it's flocculent sediment, and therefore introduce phosphorus that way through the resuspension mechanism. But it's kind of ironic, since they're intended to create the aerobic conditions where the iron would stay oxidized and bind the phosphorus. And that's why they're put in, but they're expensive. And again, as Mike very nicely pointed out, each lake is unique, and you really need to do a diagnosis on each lake to figure out what the best approach is.
Okay, and also for the other presenters, I may direct a question to one of you, but any of you feel free to chime in if you have additional input. The next one
this is Ken. Alan's correct. If you do it right, it shouldn't be resuspending it, but it is certainly possible to do it wrong.
Okay, our next question is from Steve Landry, and it could be for Al. In a lake with multiple seeps and upwellings of interstitial waters into the water column at various locations, should phosphorus monitoring be targeted at those locations in order to develop more accurate sediment treatment strategies and approaches, and initial P budgets for lakes and ponds?
It depends on the volume of the lake that you're dealing with and how important those seeps are in contributing the load. in general, what you'll find is that those seeps, at least in my experience, is those seeps will, whatever's coming through them will diffuse and end up becoming there will be a fairly uniform distribution throughout the lake bottom. So I don't necessarily would recommend that you apply just around those areas. In fact, if anything, if there's strong seeps and you're adding a chemical inactivant that Mike referred to, you're going to move it away from where it was, and you will no longer have a uniform cover on the bed of the lake bottom. I think I'll defer to Mike, who might have more experience in that than I do.
Yeah, this is well, this underpins the importance of doing a proper diagnosis of your lake, because it really depends on how much comes in there, what are the dimensions, and you might easily come up with multiple measures that need to be implemented in your lake. So this is it's difficult for me to give an advice on this, because it's really case specific, and you need to know how much and how much, well, how strong what is the water current there, because if it's not that strong, spread out over a large area, you can easily use an active barrier, no problem. We have those water bodies in my country where we have groundwater coming in, where we need to use an active barrier, and because the groundwater is so loaded with phosphates to keep it out, and then we make these calculations for how long will it take before we need to do a application. But it's you should calculate that and get the data, and I would always cover the entire lake, because you never know what happens on other sites. So you may have a priori a feeling, well, these are my hotspots, but take samples throughout the lake. Just go in there as objective as possible.
Okay, our next two questions are from Alex Woodall. This one could be for any of you. If a lake averages 6 feet deep, will alum be effective, considering the fact that summer boat activity is high?
Alum.
Well, I can tell you that the shallower the lake, the greater the chance for wind wave action to occur and to redistribute the alum application. In general, what we try and do in shallow lakes, depending on the morphometry of the lake itself, which makes a big difference, is trying to get it through the thaw web, the deeper part of that shallow lake. Avoid applying it where there's macrophytes, because it won't reach the lake bottom. It's going to get caught up by the macrophytes. And so again, you know, this is going to be beating a dead horse, but it's really important, as both Mike and Ken have said. By doing the diagnosis of your lake, a full lake diagnosis, you're going to get a better handle on where these applications should go and make sure you get the most bang for your buck. There's a lot of people who say, have told me so many times, Al, the last thing I want is another study. And I tell them, if you want your money to be spent wisely, it's a wise investment to do the science first to figure out where you're going to spend your big bucks for restoration. So please do the study first and figure out what's the right thing to do.
Yeah. To answer that question as directly as possible, I have had shallow lakes that have been treated and it has worked fine. It is not a matter of because you have some boats or that there's some wind that it won't have a positive effect. However, the very nice work done by Brian Huser and a bunch of folks found that the longevity of aluminum treatments was lower in shallow lakes than it would be in a deep lake, and that makes perfect sense.
Yeah, much slower. Yeah, so down to more or less 5 years. So instead of 20 years or even longer in deep lakes. Brian Huser, who's now working in Sweden, he's not allowed to do surface water application of aluminium over there, but he's now injecting it directly into the sediment. And that's another way of, if you are worried about these wind actions, it becomes more costly, but it also is highly effective, what I've seen so far.
Thank you, gentlemen. The next question is for Ken. When should you test for cyanobacteria, and how many times do you test for the same area?
Okay, well, you could have cyanobacteria at any time. So having an algal monitoring program that runs year round is nice. Budgets and weather being restricted factors, we usually don't worry about blue-greens until around June, and then it will go well into October when you've got the potential for blooms, even November. So the minimum programs that I run with folks involve monthly sampling, June through October. It's nice to get it every 2 weeks. Keep in mind that because those benthic origin blooms can pop up in the matter of a few days, you may need to look at it more often than that. But it doesn't mean you have to be doing the microscope work. You need surrogate things: chlorophyll, phycocyanin, the ratio of the two, Secchi disc, all the other things you can do quickly and easily that say, I may have a problem here, and then you do the microscope work.
The standard protocol in the Netherlands for official swimming sites is once every 2 weeks. Unless growth lake concentrations rise, then it's once every week. And they are using these fluoroprobe things to get a grip on cyanobacteria growth lake or phycocyanin, depending on the device they're having. It also allows you to, as Ken nicely explained, to make profiles, not to be surprised that suddenly a metalimnetic bloom surfaces. Yeah.
I'd be cautious about the use of the fluoroprobes, though, because as we all know, any dissolved organic matter in the water column can give you a deceptive reading.
Yes. Yeah. Again, the fluorescence isn't actually a direct measure. It's related to it, so you have to exercise some caution. But as a quick thing to tell you, do I have a possible issue? Should I be doing more work here? It's very nice. And the EPA does a great job with the cyanobacteria monitoring program in New England. And I'd encourage people to look into that because it's a very nice monitoring event.
Another possibility might be to set up a citizen monitoring group around your lake and just use lake color as an estimate of the bloom, so then you'd know whether to go out and sample or not. Yep.
And that the EPA program, although it doesn't just do color, is focused on citizen monitors. Again, that's it's a great topic of a totally different webinar. But when I started doing this work, you couldn't get a dime, not even as in-kind credit for using volunteers. Now it's hard to do it without them, and I'm glad because we get a lot more data that way.
And you get citizen ownership in the process too.
Yep. There's just no downside to it.
All right. We definitely love our volunteers and the data they collect. So thank you to all of them. The next question is from Kirsten Nelson. She would like to hear more about the influence of acid rain on internal loading and how sulfate has influenced phosphorus release. Can sampling hypolimnetic sulfate be informative in understanding internal load?
Hi, Kristen. How are you? That's a great question, and I think it can be. Obviously, the sulfate problems that were occurring with acid rain have been dissipated to a large degree over time, and it's no longer as problematic as it used to be. But certainly, there's legacies of sulfur in a lot of lakes, and so again, it gets back to that diagnosis. You need to understand how much what its presence is, what its total abundance is, and where it's forming its bonds inside the lake to recognize whether it's going to be problematic in terms of binding the iron and releasing the phosphorus as a consequence.
Okay, the next one is from Tim Sorrentino, and he asks, if cyanobacteria kills mussels, would that include zebra mussels?
Cyanobacteria that produce toxins will include toxins that can kill mussels. I can't say that I've ever seen or heard of a kill of zebra mussels from it. I don't know that it's not possible, but I've never heard of it. I don't know if anybody else has. But the one I mentioned from the Cape where the thermocline was much deeper one year than the next, that was a huge mussel kill. And somebody that was an engineer, retired engineer, a very sharp guy, also a volunteer monitor, went down and rescued a number of mussels. The mussel foot was actually sticking out of the shell, but they weren't dead. Put them in his well water and they recovered. So there was obviously some sort of nerve toxin involved there. So they can be impacted. I don't know of any reason that zebra mussels wouldn't be affected, but I'm certainly not recommending blue-green algal toxicity as a means to control zebra mussels.
I recommend, Tim, to check out the research that's being done in Donna Cashian's lab at Wayne State University. And she's looking at the interaction of microcystins and drycinid mussels. I don't remember offhand all of her results, but by all means, Kashian. And she's been doing work in this area and some very good work.
Okay.
Okay, the next question is directed to Ken, and it's from Janet Anderson. When the benthic growth starts, is it below the area where plants can grow? Will high macrophyte growth slow the benthic algae growth?
Okay, macrophytes can lower the benthic algae growth by restricting light penetration. Sometimes there are allelopathic, chemical interactions that can do that as well. But actually, in milfoil infestations, I find a lot more blue-green algae mats at the base of those where the stems and the roots are. They seem to be almost synergistic. So I think that the biggest issue is in fact slightly below the point where rooted plants grow. I mean, that's where the gliotrichia pops up from and a lot of other things, but it does not mean it cannot occur when you have rooted plants. It's probably a reasonable generalization that if you have a really serious rooted plant problem, you're probably not going to have a lot of benthic growth cyanobacteria popping up. It's not impossible, but there is that allelopathic interaction and the light interaction, which really matters a lot. So I guess the answer is yes, it's likely to be more prevalent below where the plants grow, but it's certainly possible within the macrophyte beds.
Okay, the next question is from Nancy Stetson, who I know is a volunteer monitor on a lake in New Hampshire. Her question is, in a shallow lake, 9 feet mean depth, where should sediment cores be taken? There's one deep spot at 27 feet. There is concern that high-speed boating on this shallow lake is detrimental to water quality. After a high-speed boating event, the normally clear water is highly turbid. Should sediment cores be taken in high-speed boating areas? Should water samples be taken and tested for total phosphorus after a high-speed boating event?
Okay, I'm going to jump on that, even though some of the other guys may have done more. I've actually worked quite a bit with boating impacts on lakes. It goes back 20 plus years and have written papers on it. First off, you need to have real data. Now, the statement that it's much more turbid after a weekend of boating is entirely possible. And I did have lakes where, indeed, you could watch the turbidity rise Friday night, and it would drop down a little bit overnight Saturday and go up all day Saturday and drop a little Sunday. And God help us when there was a 3-day weekend. It was bad. But generally speaking, once you hit a depth of about 15 feet, it is really hard for boats to have much of an impact. There's been some nice work done up in Maine looking at wakeboard boating and things like that. And once you're 15 plus feet down, you really don't get a lot of resuspension. So saying a mean depth of 9 feet tells me that at a 27-foot maximum, I'd like to see the morphometry of the whole lake. How much of it really is shallow? Are people adhering to good boating practices where they're, not making wakes when they're within a certain distance of shore? It's a whole lot of other factors that figure into it. But taking it on face value, you could indeed have an issue with resuspended phosphorus that way, as Alan talked about. A lot of it's going to settle back down. It's not going to be readily available, but some portion may be. And at that point, you have to actually take the samples, fractionate them, and get an idea of how much available phosphorus is being created or made available.
I think in general, again, to answer that question, certainly you want a sample from the deepest part of the lake as one of your sediment cores. And then it's just going to depend on the land use around the lake and where you think the potential sources are going to be greatest. Again, trying to take as broad a sampling approach as a sort of reconnaissance at the beginning, maybe just looking at total phosphorus to begin with to see whether there are hot spots or not. And then, as Ken said, start to fractionate and figure out where you might want to apply chemical inactivant if that's your ultimate goal here, or some other remediation procedure.
One of the tricks too, and people often don't do this, is don't just take a DO profile at your deep point. Let that probe drop right into the sediment in shallower water and see whether or not it's anoxic. Just because there's oxygen a foot off the bottom doesn't mean something's not going on.
Absolutely.
Okay, I would like to be cognizant of everyone's time. It is 11:59 and we are set to end at noon. However, we still have at least a dozen questions left. A couple couple hundred people on the webinar. So if our 3 presenters are willing to entertain some additional questions for a few more minutes, we can keep going. Or if
Perfect.
you want to sign off, we can do that.
It's fine with me.
Yep.
All right.
Absolutely fine.
Thank you very much, gentlemen. We'll keep going then. So our next question is from Janet Anderson, and she asks if increasing water temperatures, is that measuring water temperature at the metalimnion? So I think you were
referring to my graph, but that's actually at the bottom. It's where the hypolimnetic oxygen demand or the sediment oxygen demand is occurring. That's a temperature just off the bottom.
okay.
And if you notice, those temperatures were, low double digits, 10, 11, 14, 15, 16 degrees centigrade. That's not warm water, but that little bit of a change is a pretty big jump in oxygen demand and metabolic activity.
All right, the next one is from Kirsten Nelson, and she would like you guys to touch on Lake Browning's influence on internal loading in cyanobacteria.
Brownification. There are some studies from Sweden, from the lab of Lars Anders Hansson. They found some more toxic microcystis popping up due to brownification. So in that region, they expect some more problems with cyanobacteria. How brownification would affect internal loading, that's a difficult one. But maybe Al has some thoughts about that.
Well, there's a pretty strong humpback relationship between the dissolved organic matter entering into lakes and primary productivity, because the brownification brings nutrients with it. So if the lake is nutrient limited, you'll get an increase in primary production up until the point where the light transmissivity declines to the point where there isn't enough light for photosynthesis. So if your internal loading is related to organic matter from primary production that's settling down and decomposing at a very high rate of brownification, you're going to get less primary production, less organic matter, and less internal loading. But that's going to be over a very long time period. And my guess is you're thinking about shorter time periods, in which case I think there are other factors that are going to be much greater than brownification that's going to influence your lake's internal loading. But over the long term, certainly looking at that humpback relationship will give you an indication of future potential of internal loading in your lake.
Okay, the next one is from Rick Van Driesen, and the question is, how does the spring temperature affect the bottom temperature in the summer and the fall?
Well, the spring temperature is not going to vary wildly. It's just when the lake stratifies, what's the temperature of the water that's been trapped at the bottom. So if you have a warm spring that has gone on for a while without actually getting stratification, lots of wind and so on, the bottom water may be warmer. The actual temperature in the spring will have a certain amount of variability, but it's not just the temperature, it's wind, the morphometry of the lake, flow inputs, all the other things that combine to give you a different level for the thermocline in any given year.
Ken, will that temperature influence the aconite germination to form the algal bloom?
It should, yeah. I don't know if that 4 centigrade degree difference that makes such a huge difference to hypolimnetic oxygen demand is enough to make a big difference to aconite generation. That I don't know, but it could. And certainly, the warmer it is, the more likely the generation, or the faster, yeah.
Okay, the next is from Peter Osterman. Do planktivorous fish impact algae community in a way that can lead to competitive advantage for cyanobacteria?
I missed the first few words of that.
Do planktivorous fish impact algae community?
Got it. Yes, this is a huge thing. Maybe not the next paper, but one I really want to get out is I've been working with aluminum treatments in a number of lakes that have alewife populations. And alewife just decimate the zooplankton, whether they're sea run or they're a permanent, landlocked population. Whatever, if you don't have a good population, a high population of large-bodied cladocerans, Daphnia being the classic, you're going to get the maximum amount of algae you can get for whatever level of fertility you're at. So if you control the phosphorus and you drive the fertility down, you may be just fine. You won't necessarily have bad blooms, but you're going to get more algae than you would if you also had a really nicely functioning biological food web. And alewife are a very hot topic in a lot of New England lakes, particularly Maine. Again, it's a natural function for them to run in from the sea, spawn, have a gazillion little alewife for the summer that just wipe out the zooplankton. But in the lakes that we've done aluminum treatments like that, I see the clarity go down from 8+ meters in the spring to 3 or 4 in the summer. Now, they're not unhappy with that. But lakes that don't have alewife stay at 6 to 8. They don't get the extra algae. And it's not blue-green algae, and that's the key, is that having the low phosphorus really controls the blue-greens, but having a good zooplankton population, a favorable one, will also tend to keep the algae down even further.
Okay, this one is from Michael, and it's from Steve Landry, or Mike from Steve Landry. Could you speak to the potential downsides of hypolimnetic withdrawal and aeration relative to the cost, noise, and area required to install and maintain devices in tightly populated lake, pond, shoreline areas?
Hi, Steve. Well, that's a lot of things you want me to address here. The different devices, they all come with different costs, and of course, there are quite some engineering and installation costs there. The oxygenation system I presented from Lake Oudekerkerplas, it costed over 2 million euros to install. The maintenance of it, of course, divers have to go there once in a while. That also is an additional cost. I have no idea how much because the company is not telling that. I've asked about what are the costs for pure oxygen, and they said, well, maybe 10, maybe 15,000 euros, but maybe it could even be more. So depending on the system which is being implemented, depending on the size of your lake, and you have to run them continuously, you may come up with quite substantial costs there. And for oxygenation, hypolimnetic withdrawal, you need to build a pipe, but you don't have these costs of having generators running, having pumps running, having to add materials there. So that will definitely be cheaper. But there's a big but there. This water contains a lot of reduced substances and probably needs to be treated. And there are additional costs that need to be taken into consideration. Yeah, again, this is case specific, but these things are, well, usually not the cheapest ones. So if you would compare, and it really comes down to your diagnosis. So your diagnosis also includes a cost-benefit analysis, and in that one you also are comparing different possible interventions. If the case of Lake Raubach, for instance, internal load was a main issue there. Then you have different possibilities. Dredging, it would have been 10, 12 times more expensive than adding phosphorus for that lake. Sand capping would have been even more expensive because of the transportation cost. So for each case, it's very good to make these calculations and also have these potential alternatives next to each other. Of course, costs will include the expected longevity of the intervention as well. How long do I need to run the stuff off oxygenation? Probably continuously. For chemical fixation, maybe you need to repeat this once in a while after a decade or 15 years or whatever. So all these factors together will give you a clear picture about what will be most profitable to do in your specific case. But it's very difficult to point that this will cost that much per square meter because each lake is unique, and for substances to add it, the dose depends on how much needs to be inactivated. How much oxygen you need to put in depends on the sediment oxygen demand, how much organic matter is in there. And so all these aspects need to be taken into consideration, and then, well, make the calculation, and you will find out what will be economically most profitable for your lake or feasible.
This one is for all of you, from Scott Schuler. How many years of data are needed for good diagnosis of phosphorus loading?
Needed or preferable?
Any is possible.
It's a tough one. I know, and I've done a fair amount of work in recent years in Maine, and I love their approach. They look at all the historic data, and there's often a couple decades worth of sporadic data, and then they require 1 year of really intense data collection so that you can put it all in context. Now, any 1 year you pick may not be representative of the range of conditions, probably won't be, and it may not be the average. So long-term is good. A lesser intensity monitoring program carried out for more years is probably better than a high intensity one carried out for a short time, but how long can people afford to wait before you do something? So there's a lot of trade-offs there. Most lakes that have the wherewithal to do something, that have had that level of interest by the people that live around them, have some kind of monitoring data that give you a feel, and then you can tailor make a 1 year program to get it, fill the information gaps, and provide at least a reasonable approximation of loading and do that type of analysis that Mike's talked about.
It's a great answer.
Fully agree.
All right, this one is a little bit long. It lays out a scenario. It's from Thomas Duffy. Our lake has 4 tributaries that are 10 to 20 times higher in phosphorus input due to cow farm drainage. The lake is lowered 5 feet during the winter where the heavily laden muck is exposed or available. What would be the best methods for controlling phosphorus release, and can alum be useful in a lake in dry condition or in water less than a foot deep?
If you have that kind of an external load, you really have to look at that first before you're dealing with the internal part. I have a system in Massachusetts where we treat the 2 inflowing tributaries to a lake from late May through early July, basically Memorial Day to 4th of July, with aluminum to clean up the water entering the lake, which also then gives us a certain amount of inactivation in the sediment when the stuff settles. And that tends to get them through the whole summer with reasonable conditions. I mean, it's an urban lake. Their typical water clarity had been 4 or 5 feet before. We now average 12 to 15 for the summer. People are happy with that. It works. But if you've got that kind of an external load, you're going to be into a constant maintenance issue. You can't expect to do one thing in the lake and have it work.
As Ken says, it's always best to treat the external load first. Otherwise, you're going to be constantly treating the symptom of internal loading in your lake. So do it as a phased kind of response if you can, if you have the ability to do that. If you don't, and you just want to continuously to treat the symptom instead of the disease, then you can apply alum in a foot of water. It won't have any effect if there's no water above it at all. But ultimately, that's not the best management strategy. Getting rid of or trying to minimize the external loading first is definitely the right approach.
You have to decide at what point are you doing rehabilitation and what part are you just doing maintenance. And sometimes you don't have a choice. The one I mentioned in Massachusetts is an urban lake. It's 50 times, the watershed's 50 times the size of the lake, and it's completely developed. So it's an urban lake. We acknowledge we have to do maintenance.
Oh, sorry, Mike, were you going to add to that?
No, I fully agree. In these cases, you should definitely try to reduce your external load. Otherwise, it's, how do you say it, mopping while the faucet is still running.
All right, this next one for any of you from Rick Van Driesen. What effect do increasing chloride concentrations have on phosphorus releases and cyanobacteria blooms?
Chloride?
Yes.
So like road salt, is that the concern here?
I'm guessing that's the case. Yeah, that's what we see a lot, at least in the Northeast for sure. And I know beyond that.
It's interesting. So, in a lot of lakes that no longer turn over because of that concentration gradient of salts in the Hypolimnion, the salts are heavy and they'll settle down to the bottom. we get high phosphorus concentrations that are being internally loaded, and they stay in the hypolimnion. In this case, it's a good thing. You don't want it getting up into the upper water surface because it's going to stimulate algal blooms. So what you need to do in those circumstances is you don't want to reduce your chloride concentrations first, because that'll cause the lake to turn over. So you want to make sure you somehow get rid of your phosphorus, perhaps through chemical inactivation or hypolimnetic withdrawal, as Mike was saying, and then deal with the chloride concentrations thereafter. But as far as chloride itself influencing the internal loading directly, I'm not aware of that. It's just this indirect relationship between the two in terms of changing the lake's density and its ability to turn over.
I agree. Yep.
Okay, for any of you, are there concerns about bioturbation after a sediment cap is applied and for reintroduction of phosphorus from the sediments below the cap?
We've done those studies, exact studies. If you're interested, I'll be happy to send you the research. But very shortly, what we see is that for alum at least, I can't speak to phoslock, I'll leave that to Mike. But for alum, what they get is a smothering effect for the first year, and the benthic macroinvertebrates tend to die off in pretty significant numbers. But by the second year, the benthic invertebrate community comes back, and they have a greater evenness and diversity afterwards, which is a good thing. And their impact on the internal, the effectiveness of the alum to stop internal loading is relatively minimal.
The bioturbation is generally considered a good thing also for active capping materials, because it will mix in the material more rapidly in the communicating sediment depth. Of course, the most difficult thing about estimating a dose is to get a good grip on what is the communicating sediment depth. Is it 5 centimeters? Is it 10 centimeters? Is it 20 centimeters? So if your communicating depth is 20 centimeters and you only have targeted 5 centimeters, when bioturbation starts to mix in, you have underdosed. But if your dose is right, then it's fantastic, of course. And then it comes to what your product is doing with the organisms that are causing bioturbation. Of course, I can already mention Brian Husser's work with alum, and he also showed that carp, for instance, really can increase the communicating sediment depth, but also by doing so impact the longevity of the alum treatment, reducing it. So there are so bioturbation will have an effect depending on what's being used, but also how well you're able to get your dose right. But bottom line is, it's not a negative thing, to be honest.
Okay, this one is from Rebecca Jascott. How do you determine the natural trophic status of a lake when developing a plan to reduce P loading? Is a lake that is eutrophic due to internal loading considered to be in its natural trophic state?
When I do a model, lake loading response model, and there's lots of other ones like this, a simple spreadsheet model, but when we look at what the natural state for the lake would be, and keep in mind a lot of these lakes were artificially created, so there is no natural state, but where you're doing that, you take out all the human development in the watershed, so the loads are now based on forest and wetland, and I usually drop the internal load by about 90%. So we don't count the internal load as part of the natural state. Of course, the reality is there are naturally eutrophic lakes. Florida's got plenty of them. That's where we mine all the phosphorus for fertilizers. So you really have to look at the lake and the watershed to make that decision as to what you're going to count as natural or not natural.
And of course, some lakes have natural internal loading that hasn't been due to cultural eutrophication.
Correct.
But I think, Ken's approach is certainly the most parsimonious one.
Okay, another one from Rick Van Driesen. Is anyone aware of long-term measurements of atmospheric phosphorus available in the Northeast?
Well, there's certainly databases for how much comes out of the atmosphere. I couldn't tell you offhand what fraction is available. It's generally not a big problem. If you just have a lake that's atmospherically loaded, it's usually in pretty good shape.
All right, I just want to do a time check again. Are all three of you still good?
I actually have to sign off. I apologize, Amy, but
Okay,
thank
no
you very
worries.
much. Thanks to Ken and Mike for some great entertainment and some great education in the process.
Well, thank you, Amy, for
Thank you.
moderating. They'll be looking for you for the next presidential debate.
All right, then. You guys were very easy, not at all compared to the presidential debate. Thank you all for your presentations. And again, this webinar is recorded and I will be sharing the link to the video in the next day or two. So thanks for attending and thank you to all of our presenters. Everything was excellent.
Take care, everybody.
Yeah,
Thank
thanks.
you very much.
Thanks. Take care. Bye-bye.
Bye-bye.